Okay, hello everyone. My name is Ojaswi, and this is the third chapter that I'll be trying to finish. The chapter is Biomolecules. It is a very important chapter because it is like your building blocks for the rest of your life in life sciences. It's like the bricks that are used to construct the whole building. So it's a very important chapter. It's also one of the scary chapters because a lot of students don't prefer it. So we'll try to take all of the important content from the chapter without getting scared. The first thing about the chapter is that you should not be scared of it, and only then you can master it because it's intimidating for some people. So you just don't have to get intimidated, and that's the key. Try to live the content. Like whatever you will be reading, try to imagine it as much as possible. Whenever you are facing difficulty in imagining, you can either watch different YouTube videos for that, or you can contact me. I'll explain those parts again. But the thing is, you just don't get intimidated by the content. All right. uh, okay, I'm going to start my PhD in Region Center for Biotechnology, and these are some of the examinations that I've qualified. All right, with that, let's just start. So, questions have not come from this part. So, if you are specifically preparing for NEET, you can skip this. But I'll just read through this. So, what this content is trying to say is that whenever you try to find out what any particular tissue is made up of, for example, a plant tissue or an animal tissue, or let's say uh, you find an insect, a dead body of an insect, and you're really curious to find out the type of elements and the type of molecules that are present inside that insect. So what you have to do is first you have to take a cell, then you have to crush the cell. I mean, grind it in trichloroacetic acid, Cl3COOH. After that, you have to pass it through a cheesecloth. Pass through cheese cloth, which is a cloth-like substance. It is used to filter. After filtering, you will get two parts. One would be the liquid that comes down. That would be your acid soluble fraction because it came down with the acid, and the part that is remaining on top of the cheesecloth will be your acid insoluble part. And after getting these, you further have to go through different filtration purification methods. So I guess it must have been written somewhere here. Separated compounds, okay, analytical techniques when applied. Okay, so after that you have to apply different separation protocols to find out the different biomolecules that are present inside. If we are talking about the elements that are present in any living organism, so the major constituents are your CHON or John. So it's basically your carbon, oxygen, nitrogen, and hydrogen. Uh, Questions have not come from this part in NEET. So if you want, you can skip it, but it's just good to know like which elements are present in majority and which are your micro elements. So these four are known as macro elements and the rest are known as micro elements. All right, so let's move ahead. Um, all the carbon compounds that we get from living tissues can be called as biomolecules. So what all comes under biomolecules? When we're talking about carbohydrates, it has carbon, right? Then when we are talking about proteins, it also has carbon. When we're talking about nucleic acids, it also has carbon. When we're talking about lipids, that also has carbon. So these are your biomolecules, right? However, living organisms have also got inorganic elements and compounds. For example, there are some metals that are required to function as prosthetic groups or to function as a part of your enzymes. So for example, iron, zinc. So you also have inorganic elements. How do we know this? A slightly different but destructive experiment has to be done. So this questions have not come from this experiment. If you want, you can go through this part. All right, so let's just try to see which all elements are present. So in our body, we have sodium, potassium. These are mostly used as your to transport across ion channels to maintain a constant balance of ions between the extracellular and intracellular environment. The tonicity, hyperosmotic, hypoosmotic, so they have to maintain the osmolarity of the cell. In those cases, sodium and potassium play a major role. Then calcium is required for many functions inside your body. It activates different pathways which are required for further downstream production of certain proteins, right? Then magnesium, water is very important. And then these ions also form compounds amongst each other, and they also play significant role in signal transmission and maintaining of osmolarity and downstream signaling. All right, moving ahead. So amino acids are organic compounds containing an amino group and acidic group. So when we're talking about any amino acid, first of all, you have to learn how to draw amino acid. It's fairly easy. So NH3 plus and COO minus. This state is known as zwitterionic form of amino acid, and this form is only observable at one particular pH. It is not observed at all pH. For example, and that particular pH is known as isoelectric point. I'll just write it, isoelectric point. So for example, if there is an amino acid which has an isoelectric point of 7, right? So if you change the pH to let's say 6, then it will not be in this form. All right. In that case, it will not be in this form. Similarly, if the pH is increased to beyond 7, for example, if the pH becomes 8, then again the form will change. And how will it change? If you're changing it to an acidic level, you'll get this form. If you're changing it to a basic level, you're changing it in to this form. And this particular form will only be observed at your isoelectric point, which is known as Pi. This will be observed when the pH is greater than Pi, and this will be observed when pH is less than Pi. All right. And this Pi can be anything. It can be 6, 7, 8, depending on the type of amino acid. And why does amino acid vary? It is because of this particular functional group. So different amino acids have different functional groups. All right. So let's just go through the text first. Amino acids are organic compounds containing an amino group, which is this one, and an acidic group, which is this one, on the same carbon. That is the alpha carbon. So this is the alpha carbon. Hence, they are called alpha amino acids. They are substituted methanes. So, this compound actually comes from your methane, which is CH4, right? These are there are four substituent groups occupying the four valency of the carbon. These are hydrogen, carboxyl, amino, and a variable group known as R group, like this one. Okay, based on the nature of R group, there are many amino acids. So the R group can either make an amino acid basic, acidic, or for example, it can also contain sulfur. So it will make the amino acid a sulfur containing amino acid, etc. However, those which occur in proteins are of only 20 types. So in our body, the naturally occurring amino acids are 20 in number. The R group in these proteinaceous amino acids could be a hydrogen. So this R group can either be hydrogen. The amino acid is called glycine. Okay, I'll just rub some of this off so that it will be, I can write more relevant content. All right, so when your R group is hydrogen, it is the simplest amino acid. Simplest amino acid, which is known as glycine. Okay, questions have come from this part, so it's important. A methyl group, alanine. When R is CH4, then it becomes alanine. Hydroxymethyl, that is if it's OH, then it becomes serine. 3 of the 20 amino acids are shown. So we'll be seeing the amino acids in the next slide. The chemical and physical properties of amino acids are essentially of the amino, carboxyl and the R functional groups. Based on the number of amino and carboxyl groups, there are acidic. Okay, so questions have come from this part. So let's just write it down. Which amino acids are acidic, which are basic, and which are polar, which are non-polar. These are some of the important things that you must know. So when we are talking about basic amino acids, you have two: lysine, which is represented by K, and arginine, which is represented by R. When you're talking about acidic amino acids, we have glutamic acid, right? And you have, all right, so there were some technical issues. Anyway, so I was talking about different categories in which the amino acids are divided, right? So when we're talking about basic amino acids, I said that it counts for your lysine and arginine. When we're talking about acidic amino acids, it has your aspartic acid and glutamic acid. When we're talking about your hydroxyl containing hydroxyl, which is OH, then it is serine and threonine. When we are talking about sulfur containing, it's cysteine and methionine. So these are some of the major areas from which questions come regarding characterization of amino acids, right? Alright, so after amino acids, we'll be moving to lipids. So lipids are generally water soluble. They could be simple fatty acids. A fatty acid. Okay, so there are a particular set of questions that come from lipids, and as we go through the text, I'll highlight those parts. Okay. So lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be a methyl or ethyl or higher number of CH3. For example, palmitic. Okay, questions have come from this part. Palmitic acid has 16 carbons, including the carboxyl carbon. Arachidonic acid has 20 carbon. Okay, so remember this: when we're talking about palmitic, it has 16. When we're talking about arachidonic, it has 20, including carboxyl carbon. Fatty acids could be saturated or unsaturated. All right, from this part also a lot of questions have come. So when we're talking about saturated fatty acids, they have single bond. When we're talking about unsaturated fatty acids, they have double bonds. All right, so fatty acids could be saturated without double bond or unsaturated with one or more double bonds. Okay, so unsaturated means double bonds. Okay. Another simple lipid is glycerol, which is trihydroxypropane. Okay, so question has come from this part as well. When we're talking about glycerol, glycerol has another name, which is trihydroxypropane. Let me just rub off some of the parts so that I can write more. Yeah. So, what was I saying? Yeah. So glycerol is also known as trihydroxypropane. Many lipids have both glycerol as well as fatty acids. Here the fatty acids are found esterified with glycerol. They can be monoglyceride, diglyceride or triglyceride. They are also called fats and oils based on melting points. Oils have lower melting points and hence remain as oils. Some lipids have phosphorus and phosphorylated organic compounds in them. These are known as phospholipids. This line has also come at least 4 times in the last 10 years as question. So lecithin is a phospholipid. Phospholipid. So lecithin is one example. Some tissues, especially the neural tissues, have lipids with more complex structures. So when we are talking about lecithin, it is a phospholipid, and phospholipids are your primary metabolite. I mean, at least lecithin is your primary metabolite. So what are primary metabolite? Those metabolites which are always required in a living cell. For example, in our body, all of our cells has a plasma membrane, and lecithin is a very important part of your plasma membrane. So since it is always required, so it is a primary metabolite. The compounds which the cells produce under different types of conditions like stress conditions, okay, those compounds are known as your secondary metabolites. So lecithin is your primary metabolite. Let's move on. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogenous bases. When found attached to a sugar, they are known as nucleosides. If a phosphate group is also found esterified to the sugar, they are known as nucleotides. Adenosine, guanosine, thymidine, uridine, cytidine are nucleosides. Adenylic acid, blah blah blah, are your nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only, and DNA and RNA function as genetic materials. So over here, I'll tell you something. So what happens, for example, in case of your nucleotide, I'll tell you. This adenine basically attaches with thymine of another sugar over here. Right, so this whole structure is repeated over here because it's an anti-parallel structure. The DNA is an anti-parallel structure, right? So this type of bond is known as hydrogen bond. This phosphate group attached to your sugar, this bond is known as phosphodiester. And from this OH, another sugar molecule would be attached via this linkage. So that is known as your glycosidic. So whenever one sugar, when we are talking about sugar to sugar bond, it is known as glycosidic. So in your nucleotides, you see one glycosidic bond. When we're talking about bond between phospho group, phospho group and sugar, it's known as phosphodiester. So your nucleotide will have a glycosidic bond, it will have a phosphodiester bond, it will also have a hydrogen bond. So nucleotides will have three types of bonds. All right. When we are talking about lipids, so they have just normal your covalent bonds. Okay. This is the phospholipid lecithin that we talked about in details. This is your glycerol structure. It is a very important structure. You will keep seeing it as you know, go higher in life sciences. Then this is your triglyceride. All right. In case of your nitrogenous bases, which is ATGC. So how I have remembered, I'll tell you that. Like which all come under adenine, which comes under which comes under purine, which comes under pyrimidine. So when we are talking about purine, I've remembered it as pure, and the school that I have studied in till my 10th was AG Church School, Asansol. So AG is pure, and the other two, which is TNC, they are pyrimidine. Since there is only two options, either purine or pyrimidine, and pure is already taken by AG, so the other two comes under pyrimidine. That's how I remember. All right, and also pure structures have more. So whenever we are talking about two ring structure, they will come under purine, and similarly, one ring structure will come under pyrimidine. So let's move on. Okay, now we will be talking about primary and secondary metabolites. A lot of questions have come, like literally a lot of questions have come from this chart. All right, so let's just first read. The most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure and, if possible, synthesizing them. If one were to make a list of molecules, such a list would have thousands of organic compounds, including amino acids, sugars, etc. For reasons that are given in section, we can call these biomolecules as metabolites. In animal tissue, one notices the presence of all such categories. These are called primary metabolites. However, one analyzes plant, fungal, and microbial cells, one would see thousands of compounds other than the primary metabolites. Example, alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, and gums and spices. So these are all your secondary metabolites. This is an important line, which all compounds come under secondary metabolites. While primary metabolites have identifiable functions and play known roles in the normal physiological processes, we do not at the moment understand the role or functions of all of the secondary metabolites. However, many of them are useful to human welfare. So the secondary metabolites that are produced by different bacteria or fungus or other organisms that are useful for us, right? So and they are the secondary metabolites in all those organisms. So this is the list of them. So when we want pigments, the secondary metabolites are carotenoids and anthocyanins. When we're talking about alkaloids, it's morphine and codeine. Terpenoids, monoterpenes, diterpenes. Essential oils, lemongrass oil. Toxin, abrin and ricin. So this is important. Questions have come from this, this. Then lectins, concavalin A. I've seen questions from this. Drugs, vinblastine, curcumin. So these are drugs that are used as anti-cancer drugs. So this is also important. Polymeric substance like rubber, gum, cellulose, etc. etc. Some secondary metabolites have ecological importance. Okay, let's move ahead. So this is a very, very, very important chart. All right. Moving ahead. Let's talk about some biomacromolecules. So there is one feature common to all those compounds found in the acid soluble pool. So remember, in the very first slide, we divided the first, we ground the cell. Then after that, we in trichloroacetic acid. After that, we passed it through the cheesecloth, and then we got two fractions. One that passed through the cheesecloth, which was acid soluble, and the other that did not pass the cheesecloth, which was acid insoluble. Right. So the acid insoluble fraction has four types of organic compounds, which is proteins, nucleic acids, polysaccharides, and lipids. So a question can come that select the options which are present in the acid insoluble fraction. So over there you will have to tick protein, nucleic acid, polysaccharide and lipids. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 daltons. That means they are heavy compounds, right? And above. For this very reason, biomolecules, that is chemical compounds found in living organisms, are of two types. One, those which have molecular weights less than 1000 dalton and are usually referred to as micromolecules. So when we're talking about micromolecules, their weight is less than 1000 dalton. Okay. While those which are found in the acid insoluble fraction are called biomacromolecules. So anything that comes above 1000 Dalton would technically mean it's a macromolecule. The biomolecules in the insoluble fraction, with the exception of lipids, are polymeric substances. So lipids are monomeric, but the other three, which is your carbohydrates, proteins and nucleic acids, are polymeric. So question has come from this part. When we are talking about polymeric biomolecules, we only mean nucleic acids, carbohydrates and proteins. Okay, so then why do lipids, whose molecular weight does not exceed 8000 Dalton, come under acid insoluble fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles, which are not water soluble. So because they're not water soluble, because they're lipids, so instead of coming down with the acid soluble part, they remain on the top with the other biomacromolecules. So even though lipids are around 8,000 daltons in weight and should technically fall under micromolecules, but because they do not come down with the acid soluble fraction, that is why they are put under the macromolecule section. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not, but since their molecular weight is around 800 atoms, they are not strictly macromolecules. So the acid-soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid-insoluble fraction. Together they represent the entire chemical composition. In summary, if we represent the chemical composition of living tissue from the point of view of abundance and arrange them class-wise, you observe that majority of the questions have not come from this part, but it's interesting to know that majority of the weight of our cells is by water. After that, proteins occupy a major part, after which carbohydrates, then lipids. No, sorry, I made a mistake. Just give me 1 second. Yeah. So water comes first. After water comes proteins, obviously, because proteins are the things that make the cells work. After proteins comes your nucleic acid, because proteins are coded by nucleic acids. After nucleic acids comes your carbohydrates, after which comes your lipids, and lastly ions. Although ions are very small in percentage, but they play a huge role in the proper maintenance and working of your cellular machinery. So let's first talk about proteins. If I'm drawing the structure of protein, it will be N. So the first is N-terminal. N-terminal comes first. Then amino acid 1, amino acid 2, amino acid 3, amino acid 4, so on till amino acid N, after which comes your C terminal. C terminal. Alright. So proteins are polypeptides. They are linear chains of amino acids. So mark this sentence very clearly. They are linear chains of amino acids linked by peptide bonds. Okay, so in the question, they may give you something like proteins are made up of monomers known as peptide monomers. So there's it's not peptide monomers, it's amino acids. All right, so don't get confused between the wordings. And the bond that is present between two amino acids is known as your peptide bond. All right. Moving ahead, each protein is a polymer of amino acids, as there are 20 types of amino acids: alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer. That means any amino acid can come and join depending upon its sequence, right? And since any amino acid can come and join, it forms a heteropolymer. Homopolymer has only one type of monomer repeating n number of times. So in general, the proteins that are present inside our cells are heteropolymers. That means random arrangement of different types of amino acids depending upon the genetic coding that was there for that protein. All right. This information about the amino acid content is important, as later in your nutrition lesson you will learn that certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. All right. Questions have not really come from identify the essential amino acids or identify the non-essential amino acids till now. But if you want, you can look it up and you can learn it, because it might come in the future. All right. So the latter are those which our body can make while we get essential amino acids through our diet or food. So when we're talking about non-essential amino acids, it's not that it's not essential. It's just that our body is capable of manufacturing these amino acids on its own. So we don't have to take these amino acids from external sources. But essential amino acids are those amino acids that our body cannot produce on its own. So we have to take it from outside sources like plants and animals. So proteins carry out many functions in the living organism. Some transport nutrients across cell membranes, some fight infectious organisms, some are hormones, some are enzymes, etc. This is an important statement. Question has come from this statement. So collagen is the most abundant protein in animal world, and rubisco is the most abundant protein in the whole world. So when we're talking about just the animal world, then the highest amount of protein is present in the form of collagen. And when we're talking about the whole world, let me just rub it and write it again. So when we're talking about the whole world, then the most significant protein becomes your rubisco, right? And uh, this is important. So is this. So questions have come from this table many a times. So you have to learn that collagen is what? Collagen is the intracellular ground substance that is present in the tissues. Trypsin is an enzyme, and all of these are proteins. First of all of these are proteins. All right. Insulin is your hormone. Antibodies fight against infectious diseases. This has come, this has come, and this has come. Receptors are present on your cells. So what happens is, for example, there is a signaling molecule, right? So let's just imagine that the signaling molecule is calcium, for example. Then calcium will come and join the receptor. That receptor is a protein and it is present on the cell membrane of your cell. So this calcium will come and bind the receptor. This receptor will then in turn give signaling to the cell, and a lot of different enzymes and proteins will get activated and carry that signal further to your nucleus, to the DNA level, so that the transcription machinery understands which proteins now needs to be synthesized more. All right. And finally, GLUT4. GLUT4 enables glucose transport into the cells. Let's move to the next slide. All right, so let's talk about polysaccharides, like your sugar. So the acid insoluble pellet also has polysaccharides. So once again revising, so what was the acid insoluble pellet? Any molecule that was not passing through with the acid from the cheesecloth and which had molecular weight greater than 1000 Dalton. Okay. So, as another class of macromolecules, polysaccharides are long chains of sugars. They are threads containing different monosaccharides as building blocks. So, when you're talking about polysaccharide, the building blocks are monosaccharides. When we were talking about polypeptide, over there the building block was amino acid. Okay. Cellulose, for example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. So when we're talking about cellulose, the monomers are glucose. Alright, over here I'll just write monomer. Then cellulose is it's a homopolymer. Starch is a variant of this, but it is present as a store of energy in plant tissue. Animals have another variant called glycogen. Now this is important. Inulin is a polymer of fructose. So when we're talking about inulin, not insulin, inulin, it is a polymer of fructose. Okay, in a polysaccharide chain, say glycogen, the right end is called the reducing end and the left end is called the non-reducing end. So reducing and non-reducing are important parts again. It has branches as shown in the form of cartoon over here. Starch forms helical secondary structures. This is important question has come from this part. Starch forms helical secondary structures. In fact, starch can hold iodine molecule in the helical portion. The starch iodine is blue in color. So starch gives you the blue color because of its helical nature, right? So starch can bind iodine because it is present in the helical secondary structure, right? Cellulose does not have a complex helices and cannot hold I2. I mean iodine. So cellulose does not give iodine staining because it does not have any complex helices like starch does. So plant cell walls are made up of cellulose. Paper is made from plant pulp, which is cellulosic. There are more complex polysaccharides. For example, glucosamine, N-acetyl glucosamine, etc. Exoskeleton of arthropods, for example, have a complex polysaccharide called chitin. So when we are talking about chitin, it is also a polysaccharide, right? It's not a protein, it's a polysaccharide. These complex polysaccharides are mostly homopolymers. Let's move ahead. Now we'll be talking about nucleic acids. So the other type of macromolecules that one would find in the acid insoluble fraction is called nucleic acid. These are polynucleotides. Together with polysaccharides and polypeptides, these comprise the true macromolecular fraction. The building block is a nucleotide. So we have seen the structure of nucleotides a few slides ago, right? So a nucleotide has what? It has a sugar, it has a nitrogenous base, and it has a phosphate group. And it has three types of bond, which was hydrogen bond, glycosidic bond, and phosphodiester bond. Phosphodiester, right? So a nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is a monosaccharide, and the third is a phosphoric acid. Heterocyclic compounds, say they mean nitrogenous bases. Okay. The heterocyclic compounds of nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymidine. Adenine and guanine are substituted purines, while the rest are substituted pyrimidines. The skeletal hydrocyclic ring is known as purine and pyrimidinic. The sugar found in polynucleotides is either ribose or 2-deoxyribose. So when we're talking about RNA, it has ribose, and we're talking about DNA, it has deoxyribose. Deoxy in the sense that over here, when OH and OH is present, this is ribose, and when your OH is removed from the, so this is your 1 prime, 2 prime, 3 prime, 4 prime, and then there is another carbon over here, which is your 5 prime, right? So what happens is when your 2 prime position does not have an OH, it is known as deoxyribose. Deoxyribose. Alright, let's now talk about the structure of the proteins. So proteins are, as mentioned earlier, heteropolymers. This is an important word. They are not homopolymers, they are heteropolymers containing strings of amino acids. Structures of molecules means different things in different contexts. In organic chemistry, the structure invariably refers to the molecular formula. Organic chemists always write a two-dimensional view, blah blah blah, physicists blah blah blah. The sequence of amino acids, that is the positional information in a protein, which is the first amino acid, which is second, and so on, is the primary structure of a protein. Okay, this whole line is important. Questions have come from this whole line. So let me just first rub some of this off so I have some space for writing more. Yeah, so what it's saying is that when we are representing a protein as this N, then first amino acid, then second amino acid, then third amino acid, then so on, and then finally your C terminal. This type of structure is known as your primary structure. Okay. So the primary structure basically represents your sequence or sequence of amino acids. Primary structure. All right. The protein is imagined as a line. As you can see, it looks like a line. The left end represented by the first amino acid. So this is your first amino acid. And the right end represented by the last. So this is your last amino acid. Alright, a question has come from this particular statement, so kindly try to understand. Let's move ahead. So this is your polymeric structure. The primary structure, as you can see, looks like a line. The secondary structure, you either get a helix or a coil structure or a beta pleat sheet. All right. In your tertiary structure, what happens? Disulfide bond starts forming, and the structure becomes 3D. Then finally the quaternary structure, and quaternary structure of a protein is actually the functional structure. So whatever function the protein plays, it's because of its quaternary structure. Alright, so the first amino acid is also known as the N-terminal amino acid. The last amino acid is the C-terminal amino acid. A protein thread does not exist throughout as So this is just when it's first formed, it is taking the primary structure, but within milliseconds or even faster than that, this primary structure gets converted and folded into your tertiary structure. So it does not always remain as the primary structure. It gets converted super fast into your tertiary structure. A protein thread does not exist throughout as an extended rigid rod. The thread is similar. The thread is folded in the form of a helix. Of course, only some portions of the protein thread are arranged in the form of helix. In proteins, only right-handed helices are observed. This is an important statement. That's whenever we are talking about proteins, in proteins only right-handed helices are observed. Other regions of the protein thread are folded into other forms known as secondary structure. In addition, it can also form tertiary structure, which gives us a 3D view. So when we're talking about tertiary structure, it gives us a three-dimensional view of the protein. It's absolutely necessary for the proteins to have a biological activity. So the tertiary structure is very necessary for the protein to perform its functions. If the tertiary structure is not there, then the protein will not be able to perform its functions. So moving ahead, some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides are arranged with respect to each other is the architecture, otherwise known as quaternary structure. This part has also come in your NEET. So it's adult humans hemoglobin consists of 4 subunits. 2 of these are alpha type and the other 2 are beta type. Alright, this is important. Now let's talk about enzymes, because a lot of questions have come from enzymes as well. So almost all enzymes are proteins. When we are talking about almost all, that means that when we are talking about enzymes, it can also, it's mostly proteins, but it can also be RNA. When we are talking about enzymes which are from RNA, we are talking about ribozymes. Alright. So there are some nucleic acids that behave as enzymes. For example, your ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure because it's mostly protein, so it will have a primary structure. That is an amino acid sequence of the protein. An enzyme, like any protein, has a secondary structure as well, and the third tertiary structure, because without the tertiary structure, it will not be able to function. So when you look at the tertiary structure, you will notice that the backbone of the protein chain folds upon itself. The chain crisscrosses itself, and hence many crevices or pockets are made. So these, for example, if a protein is folding in such a manner, right? So this is a pocket that is formed. This pocket is basically representing your active site. The active site has a collection of amino acids, right? So over here there will be many amino acids, and these amino acids will be actually the ones that are playing the catalytic role. Okay, so one such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. Over here, my substrate, which looks something like this, will come and fit, right? Enzyme catalysis differs from inorganic catalysts in many ways, but one major difference needs mention. The inorganic catalysts work efficiently at high temperatures and high pressure, while enzymes get damaged at high temperature. Why? Because enzymes are proteins. If you give it high temperature, these proteins will break. The amino acids will start breaking from each other, and it will leave the enzyme non-functional, because for any protein to function, it needs its tertiary structure, right? So, however, enzymes isolated from organisms who normally live under extremely high temperatures are stable and retain their catalytic power at high temperature also. For example, there is a particular DNA, what is that? Polymerase. So there's a particular polymerase that is extracted from a bacteria which lives in thermophilic conditions. As a sort of activity, I want you all to find out the name of some enzymes that are extracted from bacteria or any other organism which lives under extremely high temperatures, and find out their uses in common day-to-day life that we are using as scientists. Okay, the thermostability is an important quality of such enzymes isolated from thermophilic organisms. Moving ahead, let's talk about the chemical reactions that these particular enzymes perform. So how do we understand the enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of reaction, a physical change or a chemical change. A physical change is referred to as change in shape without breaking of bonds. The most important line when we're talking about physical or chemical changes, that's whenever there is no breaking of bond, it is a physical change. When we're talking about breaking and formation of bonds, it becomes a chemical change. Another physical process is change of state of matter. For example, when ice melts to water or water becomes vapor. These are also physical processes. However, when bonds are broken and new bonds are formed, this is known as your chemical reaction. For example, this one. However, this will not come in the exam, but just for you to understand. Similarly, hydrolysis of starch into glucose is an organic chemical reaction, and this is the rate of the reaction. Rate can also be called velocity. Rates of physical and chemical process are influenced by, okay, a general rule of thumb is that the rate doubles or decreases by half for every 10-degree change in either direction. Question has not come from this part, but just looking at it, I'm just guessing that it may come. So that's why I marked it. Catalyzed reactions proceeds at rates vastly higher than that of uncatalyzed ones. When enzymes catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reaction. Okay, so when we are using the enzyme carbonic anhydrase, then the reaction is faster. But when this enzyme is not present, then the reaction becomes slower. Alright, so carbonic anhydrase, this is important, it has come in the exam. When we are talking about CO2 plus H2O becoming H2CO3, the enzyme uses carbonic anhydrase. Moving ahead, in the absence of any enzyme, this reaction is very slow with about 200 molecules of H2CO3 forming in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakh molecules being formed every second. The enzyme has accelerated the reaction by 10 million times. The power of enzymes is incredible indeed. That's true. There are thousands of enzymes, each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is known as metabolic pathway. For example, glucose to pyruvic acid. This particular metabolic pathway comes under your where. This is another your task. Find out where in which cycle uh, the glucose is converted to pyruvic acid. What is the name of that particular cycle? All right. Um, when you study respiration in 12th, you will study these reactions. At this stage, you should know that this very metabolic pathway with one or two additional reactions give rise to a variety of metabolic end products. In our skeletal muscles, under anaerobic conditions, lactic acid is formed. Again, so the formation of lactic acid after pyruvic acid, it can go to two different steps. One is aerobic respiration, one is anaerobic respiration. If it undergoes anaerobic respiration, it forms lactic acid. Under normal aerobic condition, pyruvic acid is formed in yeast. During fermentation, the same pathway leads to the production of ethanol. Hence, in different conditions, different types of products are possible. Uh, Nothing has come in NEET from this particular part as of now. So how do enzymes bring about such high rates of chemical conversion? To understand this, we should study enzymes a little more. We have already understood the idea of active site. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called a substrate. Hence, enzymes, that is proteins with three-dimensional structures including an active site, convert substrate to a product. This is the function of the enzyme. It is now understood the substrate has to bind to the enzyme. So what happens? First of all, there is your enzyme. Then comes the substrate. It forms an enzyme substrate complex. Substrate comes at active site. Then it forms your enzyme substrate complex. Then it forms your uh, enzyme product complex. Then the product goes out and your enzyme is again ready for another cycle of conversion. So it is now understood that the substrate has to bind to the enzyme at the active site. The substrate has to diffuse towards the active site. Thus, there is an obligatory formation of enzyme substrate complex. This complex formation is a transient phenomena. That means it happens for a very small amount of time and it is not a permanent structure. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. So this particular, I'll write it in yellow, this particular structure is known as your transient state structure. All right. So very soon after the expected bond breaking is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of products. The pathway must go through the so-called transition state structure. For example, over here. Now in this figure, as you can see that the initial energy of the substrate was higher compared to the energy of the product. That means this particular type of reaction is exothermic. That means upon the formation of product, heat would be given out, right? And this is the amount of energy that is required in the presence of enzyme, and a much larger amount of energy was required without the enzyme. So it's always better to spend less energy, right? So that's why when you are using an enzyme, since less energy is required, the reaction proceeds faster. Okay, there could be many more altered structure states between the stable substrate and the product. Implicit in the statement is the fact that all intermediate structural states are unstable. Stability is something related to energy. So you don't have to go through all this. This graph is all you need. This graph is what comes in NEET as well. So it's important. The y-axis represents the potential energy, that is potential energy. The x-axis represents the progress of the reaction. You would notice two things: the energy level difference between S and P. If P is at a lower level than S, like in this case, P is at a lower level than S, then the reaction is exothermic. One need not supply energy in order to form the product. However, whether it is an exothermic or spontaneous reaction or endothermic, the S has to go through a much higher energy state or transition state. The difference in average energy content of S from that of its transition is known as activation energy. So this is your activation energy, right? This is the transition state. So the difference between the energy of your transition state, so energy at your transition state minus energy of the substrate alone, is known as your activation energy. Activation energy. Enzymes eventually bring down the energy barrier, making the transition more easy. So what is the nature of enzyme action? Each enzyme has a substrate binding site that a highly reactive enzyme complex is produced. The complex is short-lived and dissociates into product. This is important. Questions have come from this part. At least I have seen, I think, 2 questions that have come in the past 10 years from this paragraph in the form of arrange the events. Like how does it happen? First, the substrate comes, binds to the active site of the enzyme and so on. So the, what can I say, the flow chart of the steps. All right, let's move ahead. So what are the factors affecting the enzyme activity? The activity of an enzyme can be affected by change in temperature and pH. So this is what is important. So as you increase the pH till a certain point, the enzyme activity will increase, after which upon further changing the pH to either left or right, I mean either making the pH acidic or basic, whatever you do, after a certain point the tertiary structure of the protein will start changing because of the change in the individual amino acids, right? As I said that each amino acid has a Zwitter ionic form at its isoelectric point. So if you change the pH to either higher than its pi or lower than its pi, what happens that the amino acid becomes either acidic or basic, right? And if the amino acids change their property from acidic to basic, then the tertiary structure will disrupt, right? So the enzyme activity will decrease upon changing the pH to either of the two sides. Similar reaction is seen in case of temperature, because what happens, if you increase the temperature or decrease the temperature too much, the tertiary structure of the protein will change. Okay, and concentration. This particular graph is important. It's known as your Michaelis-Menten graph. So I'll just explain you an equation which is known as V naught is equals to V max into S by Km plus S. Over here, S is your substrate concentration. How much substrate you are putting. V max is the maximum over here, is the maximum velocity of the reaction. And Km is the affinity of your protein towards the substrate. So for example, let's say, what can I say, I have masala tea and I have elaichi tea with me. So if I like elaichi tea much more, and if I have this masala tea, since I like elaichi tea much more, I will not be giving it to any other person that asks me for it. But masala tea, if I got masala tea from someone, I will give it to the next person as well if the person asks me. So for me to catch the masala tea and to let it go was easier, right? So my Km was low towards the masala tea, right? So when your Km is low, when the Km of the enzyme is low for its substrate, what happens is it is easier for the enzyme to capture the substrate and transform it to the product and let it go. Okay, so its affinity for it is low and hence the reaction is faster. I will be able to take a lot of masala tea from this person, give it to the next, take it, give it, take it, give it. With the elaichi tea, as soon as I take it, I'm not ready to let it go. So my hand is occupied, right? So for me to process this, it will take a longer time. So when you're talking about the enzyme activity, right? When we're talking about the enzyme activity of any particular enzyme, in that case, lower the Km, that is the attachment of the enzyme towards its substrate. Lower the Km, better the activity, right? So this is a concept that I wanted you to understand. In case it is still not clear, then please. Alright, so due to some too much of tapping onto my writing pad, there was some issue. So let's just read the parts that are still remaining. So we have discussed about temperature and pH. We have discussed about concentration of substrate from this particular graph, right? So what happens as you increase the concentration of your substrate, the enzyme activity increases, right? Because it is getting more amount of substrate, so it is converting more of the substrate to the product. So initially it increases, the velocity of the reaction increases till a certain point, after which it becomes constant, because whatever enzymes there were, for example, there were 100 number of enzymes and there was initially one glucose. So one glucose was converting, then 50 glucose converted, then 100 glucose. So all of the enzymes are now occupied with the 100 glucose. There's no extra enzyme left. So if you increase the amount of glucose from 100 to 200, then also the rate will not change because the enzymes are only 100 in number, right? So yeah, so that's basically it. As you increase the concentration of the substrate, the rate of the reaction would increase till a certain point, after which all the enzymes get saturated. Okay, So with the increase in substrate concentration, the velocity rises at first. The reaction ultimately reaches a maximum velocity, which is not exceeded by any further rise in concentration. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzymes to bind with the additional substrate molecules. The activity of an enzyme is also sensitive to the presence of specific chemicals. Over here, we are talking about inhibition. So if you want to inhibit the activity of any enzyme, what you will do? For example, if there is an enzyme which looks like this and its substrate looks like this. So the substrate will go and bind the active site and get converted to a product. But if I don't want that product because it is damaging me somehow, so what I can do is I can make a replica of this substrate which looks like this substrate but is not the actual thing. Okay, I have synthesized it and its properties are different. So what it can do, it can go and bind over here, and once it goes and bind over there, so the original substrate will not be able to bind. This type of process is known as competitive inhibition. These two are competing to bind to the active site. Competitive inhibition. So, when the binding of the chemicals shuts off enzymatic activity, it is known as inhibition, and the chemical is known as the inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity, it is known as competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with substrate for substrate binding site. Consequently, the substrate cannot bind. As a result, the enzyme action declines. That is, for example, this is very important. It has come many a times. So, inhibition of succinic dehydrogenase by malonate, which closely resembles succinate in structure. So, over here in this example, this is the enzyme which is succinic dehydrogenase. This is your inhibitor, which is malonate, and this is the original substrate, which was succinate. Okay, such competitive inhibitors are often used to control bacterial pathogens. So what happens in competitive inhibition? If you give a lot of the original substrate, which is succinate, since the amount of succinate increases compared to the malonate, which was the inhibitor. So if you give more amount of substrate and less amount of inhibitor, the substrate will eventually, win, and it will be able to bind to the enzyme and get converted to the product. So in case of competitive inhibition, if you increase the amount of substrate, you can again reach your Vmax. Okay, but in case of your competitive inhibition, I'll just write it in the next slide. Yeah, so when we're talking about competitive inhibition, the graph actually becomes something like this. This was your original graph. This was your Vmax. This is your Km. This is your substrate, right? So sorry, this is not your V. This is Vmax by 2, and this is your Km. So again, Km, which is the Michaelis-Menten constant, which I said is the attachment that the enzyme has towards its substrate. It also serves another function. So Km is basically the enzyme, Km is basically the substrate concentration at which you have achieved half of the maximum velocity. So in your competitive inhibition, what happens is that if you increase the amount of substrate, eventually you will reach the same Vmax eventually after increasing the substrate quite a lot, right? So since you have increased the substrate so much, but the Vmax is still same, so what happens that you will reach a Vmax by 2 at a later stage, in the sense that you will require more amount of substrate to reach half of the maximum velocity. So in your competitive inhibition, the Km increases. That is, the amount of substrate required to reach the half of maximum velocity increases. Alright, so questions have come from this. So I just give you a brief description. Classification, nomenclature of enzymes. Questions have not come from this part, but we'll still go through this. So how I've remembered is that these are certain classes of enzymes, right? This is class 1, class 2, class 3, class 4, class 5, class 6, some like that. So class 1 is O, then T, then H, L, I, L. O stands for oxidoreductase or dehydrogenases. Then T stands for transferases, H for hydrolases, L for lyase. That means it causes removal or lysis basically means breakdown. So lyases is like removal of groups from substrates. I is isomerases, L is ligase. Ligase serves the function of joining. Okay. So lyase and ligase are basically totally different in function. Lyase breaks, ligase joins. Alright. Then we are talking about cofactors. A lot of questions have come from this part, like a lot of questions have come from this part. So pay attention. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called cofactors are bound to the enzymes to make enzymes catalytically active. I'll just remove this so that I get some space to write. Alright, so in these instances, the protein portion of the enzyme is known as the apoenzyme. So apoenzyme, which is basically the protein. Three kinds of cofactors, apoenzyme plus cofactor. And this cofactor can be any of the three types: prosthetic groups, coenzymes, or metal ions. Cofactor can be prosthetic group, coenzyme, or ions directly. Prosthetic groups are organic compounds. So when we're talking about a cofactor which is organic, it is prosthetic group. Organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group. Questions have come from this statement. So when we're talking about peroxidase, where can I write it? Peroxidase has heme, and heme is what? It's a prosthetic group. Coenzymes are also organic compounds, but their association is only transient. That means they're not as strongly attached to the protein part as the prosthetic group is, right? Usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. For example, coenzyme NAD and NADP contain the vitamin niacin. Okay, so again, this has come in exam. When we're talking about NAD and NADP, they have the vitamin niacin. Okay, remember this. A number of enzymes require metal ions for their activity, which forms coordination bond with side chains and at the same time form one or more coordination bonds with the substrate. For example, zinc. So zinc is a cofactor for the enzyme carboxypeptidase. This has also come many a times in the exam. So remember this, that when you're talking about carboxypeptidase, you have to associate it with zinc being the cofactor. Okay. So catalytic activity is lost when the cofactor is removed, which justifies that they play a crucial role. So when apoenzyme and cofactor join, they form holoenzyme. That means the proper full enzyme, the functional enzyme. All right. So that was it. These are some extra things that I have prepared because questions have come from these parts. So these are all your secondary metabolites. In the past, questions have come from this part, so I just thought it would be useful. When we are talking about cocaine, it is derived from erythroxylum. So cocaine, just something to remember by. Heroin is derived from papaver. HEPA. Morphine is derived from papaver somniferum again, MOPA, HEPA and MOPA. However, morphine is also associated with being used in surgery. So before giving surgery, morphine is given to the patient so that they lose their consciousness, so that it's easier to operate on them without causing pain to them. Then talking about marijuana. So marijuana is from cannabis, so maca. This is how you can remember. Moving ahead, terpenoids are a class of organic chemicals similar to terpenes and diterpenes. We have read it in the slides also. Lectins are a type of proteins that can bind specifically to certain sugars and also play a role in recognizing carbohydrates. This was there in the slides, but I'm just highlighting it that concavalin A is a lectin. Alkaloids are a class of naturally occurring organic compounds that mostly contain basic nitrogen atoms. Codeine is an alkaloid formed in opium. So codeine is an alkaloid formed in opium. Toxins are poisonous substances produced within living organisms. For example, ricin. Ricin is a toxin. Okay, and this was just something that I thought you should look at before going to give the exam. So palmitic acid has 16, P16, A20. Arachidonic acid has 20. Lecithin is a phospholipid. Glycerol has 3 carbons, each bearing OH group. Alright.